Hello students, in this video we're going to look at topic 3 of the 6.2 notes. In topic 3 we're going to look at how genes are regulated in bacteria, our prokaryotic cells. And so just to remind you guys, gene regulation is the ability of cells to control what genes are being used and what genes are not being used, and then changing the expression of certain genes as needed. And so sometimes in a cell, there's certain genes that are being actively transcribed and translated, meaning that those genes are being used to produce functional proteins that are doing important things inside the cells. And some genes are not being used. And it just depends on which genes are needed. But cells are able to control when certain genes are being expressed and when certain genes are not being expressed, depending on what genes are needed and how much those genes are needed. Some genes are being expressed a lot sometimes, making a lot of a certain protein. Some genes are being expressed a little bit, making a little bit of certain proteins. And some genes, like I said, are not being expressed at all, making none of those proteins. And it just depends on what's going on and what the cell needs to be accomplishing. And so in bacteria cells and prokaryotic cells, a lot of this takes place depending on what's going on in the environment. So depending on what's going on in the environment, bacteria cells can respond a lot of times by controlling which genes they're using at any given moment. And so a good example of this is, well, in this picture right here, we're going to be talking about this a lot in this video. But this is an example of 5 genes in prokaryotic cells and bacteria cells that all together produce 3 functional enzymes. And so what's going on here is, so there's these 5 genes here. This is in the DNA of prokaryotic cells, the TRPE, TRPD, TRPC, B, and A genes. And these genes, each of these genes make has the instructions for a sequence of amino acids, which we call a polypeptide that folds up. Some of those polypeptides come together to form one functional protein because this protein has a quaternary structure. But together they form one functional protein here. Like in this enzyme here, there's 2 polypeptides that are put together here. Each of those polypeptides coded by 2 different genes. And so anyway, you guys look here, these 2 genes produce this enzyme, and this gene produces this enzyme, and these 2 genes produce polypeptides that come together to form this enzyme. And so these 5 genes right here, like I said, form these 3 enzymes. And these 3 enzymes in bacteria cells actually form a metabolic pathway, which is a sequence of chemical reactions that's ultimately producing some kind of final product. In this example, these 5 genes, the TRPEDCBA gene, and the enzymes produced from those genes are creating a molecule called tryptophan. Tryptophan is a really important amino acid that all cells need. And so it's one of the 20 different amino acids that exist. And so anytime you're making proteins, a lot of times you're going to need this amino acid to build lots of different proteins. So it's pretty important that you have this amino acid. And bacteria cells, or certain types of bacteria cells, they have genes, these genes that can actually code for the synthesis of that molecule. And so what happens though is when a bacteria cell is in an environment that has a lot of tryptophan, like there's like maybe the cell woke up today and there's like a bunch of tryptophan everywhere inside the cell and inside the environment of the cell. Well, then what actually happens is that causes these genes to then turn off or to stop being expressed. And so that's what this negative arrow represents here. It represents when there's a lot of tryptophan, it can actually start causing the repression of these genes, the expression of these genes to decrease, which if you think about it, that actually makes a lot of sense. If you already have a lot of tryptophan, you probably don't need to keep making tryptophan. And so this is an example of gene regulation where genes are being used when they're needed and then not being used when they're not needed. And that's super important for the efficiency of the cells. So by cells being able to control which genes are being used and how much those genes are being used and only using genes when they're needed and not using genes when they're not needed, that helps to conserve energy and resources in the cell. So it makes cells really, really smart and efficient by being able to control what genes are being used and how they're being used. And so this example with tryptophan and this metabolic pathway that synthesizes tryptophan, which are controlled by these 5 genes, is a great example. Again, when there's not a lot of tryptophan, these genes are being expressed, making these enzymes that build tryptophan. But if there's a lot of tryptophan, maybe there's just the cells in an environment that already has this molecule floating around everywhere, well, then it's freely available. So how about you save energy and not use time and energy to build this molecule if it's already present everywhere inside the environment. And so that's what we're going to look at in these notes is how this actually takes place. And we're going to look at this specific example in a moment here, actually. And so the goal is to kind of understand on a molecular level what's going on to actually allow these genes to be on or off. And so to do that, I'm going to actually talk you guys through this picture here. And so in bacteria cells, so again, these notes are just focused on bacteria cells, prokaryotic cells. And the next topic 4, we'll look at eukaryotic cells, and there's some similarities and differences in how our genes are regulated. But in prokaryotic cells, this is what I'm about to explain here is a common thing that we see in the DNA of prokaryotic cells. And what we have here is a situation where multiple genes are actually next to each other in the DNA, all sharing a promoter sequence. And so a promoter, just to remind you, a promoter is a sequence of DNA that RNA polymerase binds to, and RNA polymerase is going to bind to the promoter sequence and then read the DNA and make mRNA, and then that mRNA gets sent to a ribosome, and then we make a protein. And in prokaryotic cells, oftentimes we find situations where there's actually groups of genes, like not just one gene, but multiple genes all together sharing a promoter sequence. And so what that causes to happen is when RNA polymerase binds to this promoter sequence, it's going to read all of these genes all at once, and it's going to transcribe and make mRNA with the instructions for all of these genes, which will then be translated by ribosomes into these 5 different polypeptides, some of which come together to form ultimately three enzymes, those 3 enzymes that I showed you needed to build tryptophan in this picture right here. And so this is actually how those 5 genes are structured together in the prokaryotic DNA. And so the reason this is also, one reason why this is actually super beneficial is because these genes work together. So these genes are ultimately producing proteins that work together to accomplish some overall goal, which in this case is to build tryptophan, to form the metabolic pathway that builds tryptophan. And part of gene regulation is not just controlling which genes are being used, but it's also the process of coordinating the expression of certain genes together, so that sometimes when you need to do something in the cell, it might require not just one gene, but many genes all at the same time being expressed. And so gene regulation also has to do with making sure that if you want to accomplish this thing in the cell, making sure that all the genes that are needed are being turned on together. We call that coordination, the coordination of gene expression. So we're making sure that these genes are all being expressed together because we're going to need them all of those genes to do this one thing. So in this case, these 5 genes, it's actually really convenient that they're grouped together, all sharing a promoter, because these 5 genes would basically be useless without each other. Like if you just had like 1 or 2 or 4 of the 5 genes being expressed, but not all 5, well, then it kind of defeats the purpose, because you need all 5 of those genes to express to build tryptophan. And if one of those genes was not being expressed, then basically you're wasting your time, because you need all 5 to form this metabolic pathway. And so by them all being in the DNA sharing a promoter together, it's going to ensure that when these genes are expressed, that they always get expressed together. And so if one of these genes is being is turned on in the cell and actively being expressed, well, then all of these genes would be actively being expressed, because anytime one of these genes are being expressed, all of them are being expressed, because they all share a promoter sequence. So anytime RNA polymerase comes and binds to this DNA, RNA polymerase is going to read not just one of these genes, but all of these genes and make mRNA for all of them, which then will always be sent together to ribosomes to make all 5 of these proteins. So this makes sure that all 5 of these proteins are made together always, and so that we're not forgetting anyone. So that's a convenient situation here that we see happening in prokaryotic cells. By the way, in eukaryotic cells, this would never happen. In eukaryotic cells, all of your genes have their own promoter. Never in eukaryotic cells do we see multiple genes sharing the same promoter sequence. This is something that's specific to prokaryotic cells, where we see sometimes groups of genes sometimes sharing the same promoter, like this situation right here. Now, these genes, like I said, can be on or off. Like sometimes these genes are being expressed a lot and sometimes these genes are not being expressed. And it actually is controlled by this other sequence of DNA called an operator. So an operator is this common type of regulatory sequence we see in prokaryotic cells that kind of acts like an on-off switch. And so this operator, which you guys can see is kind of part of the promoter, which is usually the case. The operator is the sequence of DNA. So this is DNA, a sequence of DNA. And we say that this is a regulatory sequence. A regulatory sequence is a sequence of DNA that proteins, regulatory proteins, can bind to. So this is a sequence of DNA that a certain protein is able to bind to. And depending on whether or not that protein is here, binding to the operator, these genes are going to be either actively being expressed or not actively being expressed. And so we'll look at that in a second, how that takes place. But altogether, when you have a promoter and then multiple genes sharing that promoter and then this operator sequence, together we call that an operon, which is what we're going to be talking about a lot in this video. So operons are these situations, which again, this is only in prokaryotic cells, where multiple genes are all sharing a promoter sequence. And then within that area of the DNA, usually in the promoter sequence, there's this other sequence called an operator, which is going to serve as an on-off switch, which I'll explain right now in a moment. So that's an operon. Okay, so just there you go, right here. And so this is called the TRP operon because it controls the TRP genes. And so these are the 5 TRP genes that build these enzymes that are going to be used to build tryptophan in the cell. And so this operator, the on-off switch, hopefully it'll make sense in a moment why this is thought of as like an on-off switch. There's basically a protein, a repressor protein that's able to bind to this operator. And when the repressor protein binds to this operator, these RNA polymerase will be blocked from transcribing these genes. So there's a protein being made in bacteria cells that can actually bind to this specific operator, this sequence of DNA. So this is a sequence of DNA that a certain repressor protein can bind to. And when it binds to this operator, these genes will not be transcribed. RNA polymerase will not be able to transcribe these genes. It's going to block RNA polymerase from transcribing those genes. And that repressor protein is actually right here, if you look here. So somewhere else there's in the DNA, so this slash here means like somewhere far away in the DNA, like not part of this operon, there's a regulatory gene. A regulatory gene is a gene that codes for a protein, and it's a protein that's going to bind to the DNA somewhere and affect the expression of other genes. That's what we call a regulatory protein. So a regulatory protein is a protein that's coded by a regulatory gene. So regulatory genes code for regulatory proteins. And in the last topic, topic 2, we talked about how those regulatory proteins can be repressors or activators. Repressor proteins bind to the DNA and cause the expression of certain genes to decrease, whereas activator proteins bind to the DNA somewhere and cause the expression of certain genes to increase. But in this case, this is a repressor protein. And so there's a regulatory gene that codes for this repressor protein. And this repressor protein has a specific structural shape that allows it to find this sequence of DNA, this operator sequence, and bind to it. And when it binds to this sequence of DNA, these genes will turn off, which you'll see pictures of this in a second. Okay. Now here's the thing. So that's the gist of how these operons work. So an operon is a group of genes that work together that are all sharing a promoter sequence. And that promoter sequence and those genes, well, those genes will be on or off depending on this operator sequence and whether or not there is a repressor protein binding to it. If there's not a repressor protein binding to it, usually RNA polymerase will be able to bind to the promoter and then transcribe those genes, and those genes will be actively expressed. But if the repressor protein is bound to the operator, then that's not going to be able to happen. And so there's actually 2 types of operons, 2 main types of operons. There's operons called repressible operons and operons called inducible operons. So I'm going to explain both of them to you right now. We're going to start with repressible operons. So a repressible operon is actually the example that I'm talking about right now with tryptophan, the TRP operon. This is an operon where the genes are by default being expressed. So in the default state of this cell, these genes are just by default on being expressed. However, and that's because the repressor protein that binds to the operator, the repressor protein is by default inactive. And since the repressor protein, so if I go back to this picture here, here's the repressor protein. This repressor protein, when it's built and put together and expressed, it actually is by default in its inactive conformation, meaning that it's by default in a specific shape that makes it inactive. Like it can't do its job. It's just asleep. So we've talked about this a lot throughout the year, that proteins, like sometimes they can change shapes into their active and inactive conformation. And so this repressor protein is actually by default inactive, basically do nothing, just floating around in the cell, not doing its job. But when this repressor protein becomes active, he'll be able to do his job, and his job is to go bind to the operator and stop the expression of these genes. But by default, he's actually inactive, which means by default, this operon is by default on. Like there's no problem here. RNA polymerase can come read these genes and transcribe these genes so that these genes can then be translated and we can build these proteins. And so that's what we call a repressible operon. A repressible operon is by default on because the repressor protein is by default inactive. Okay. However, a repressible operon is able to be repressed. Okay. Hence the name repressible. It's able to be repressed, meaning yes, it's on by default, but we are able to repress it. And to repress it, all we need is a molecule to bind to this repressor protein and cause this repressor protein to change into its active conformation. And when that happens, this protein, when it becomes active, will then bind to the operator and turn off these genes. And so that's exactly what happens with the TRP operon. So in this picture here, if you guys look, like I just explained, this repressor protein is by default inactive. It's being made, but it's just doing nothing. It's in its inactive shape. And so there's no one binding to the operator. And so RNA polymerase can just freely express these genes all the time, and these genes are on. However, there is a situation that can occur where this molecule can bind to the repressor. And when it binds to this repressor, it causes the repressor to become active, turn into its active conformation, which then it can do its job, and its job is to go bind to the operator sequence and in the DNA and block RNA polymerase so that he can't transcribe these genes, and then these genes are going to turn off. And the molecule that binds to a repressor and activates it is called a co-repressor. So a co-repressor is not the repressor protein itself. It's actually usually a non-protein molecule in the cell that can bind to the repressor and cause it to change to its active conformation, causing it to then stop these genes from being expressed. That's what we call a co-repressor. So co-repressors are molecules that bind to repressor proteins and cause them to become active, which then causes them to repress the genes and cause the genes to stop being expressed. And in this specific situation, the co-repressor is tryptophan. Tryptophan, the molecule that is being made by these genes, is actually the co-repressor. So these genes code for proteins that build tryptophan, and tryptophan actually is the co-repressor that causes these genes to turn off, which if you think of, at first that seems counterproductive. Why would the thing we're making be the thing that turns off these genes? But it's actually really smart, because what's going to happen is when there's a lot of tryptophan, either we're making too much tryptophan, or maybe the cell is in an environment where there's tryptophan freely available in the environment. Like this amino acid is everywhere. Well, then it's going to start binding to this repressor protein and turn this repressor protein off and stop the expression of those genes, which is actually a perfect time. This would be a good time to turn off these genes, because if tryptophan is binding to this repressor, it probably means that there's a lot of tryptophan in the cell, which means it's now becoming maybe not smart to be building tryptophan if there's already a lot of tryptophan, so much tryptophan that it's coincidentally binding to this repressor protein. And so now we can save our time and energy by turning off these genes, because apparently there's a lot of tryptophan, which is the only reason why it would be binding to this repressor protein. And so that's what's happening with the TRP operon. Okay, so just to recap here, the TRP operon, this is a specific example of an operon, is a group of genes that together build these enzymes that are needed to build tryptophan. Okay, and these genes are by default on. And that's because the repressor protein is by default inactive, right? So on a typical day, these genes are on, the repressor protein is doing nothing. But on a day where there's a lot of tryptophan, it can bind to this repressor protein. It can act as the co-repressor. A co-repressor will then bind to the repressor protein and activate it, which will then allow it to do its job today, which is to bind to the operator and stop RNA polymerase from transcribing those genes. So now those genes turn off. And those genes will remain off until tryptophan is no longer abundant in the environment or inside the cell, which that would be a great time for this repressor protein to go back to sleep, is when tryptophan is gone. So if there's not a lot of tryptophan, this repressor protein should be inactive, and these genes should be on making tryptophan. But if there is a lot of tryptophan, it will bind to the repressor and activate it, causing these genes to turn off. So it's actually really smart the way this operon is set up. And so hopefully you guys can see why this operator acts as an on-off switch. So this operator is a sequence that repressor proteins can bind to only when they're active, causing those genes to then turn off. So that's a repressible operon. And so now I want to compare that to the other type of operon, which is an inducible operon. And hopefully this will be a lot easier now that you understand the gist of what an operon is. And so in an inducible operon, see if you can notice the difference. So here's an example of an inducible operon, which an example of an inducible operon is the lac operon. So this is a different operon. In the lac operon, there is a group of genes, 3 genes, that code for 3 different enzymes. And these 3 different enzymes have work together to move lactose, which is a sugar. It moves lactose into the bacteria cell and then breaks down the lactose into simple sugars that are digestible, like including glucose, that are digestible by the bacteria cell. So lactose is a disaccharide. It's made of 2 monosaccharides. And in bacteria cells, they have a group of genes here that will move lactose into the cell and then allow it to break down lactose, to digest lactose into simple sugars that it can use for energy. So that's what these genes do. And so they kind of work together to do that. So there's the LAC-Z, Y, and A gene, and all 3 of those genes work together to basically digest lactose. And so in this operon though, so again, those are the 3 genes working together. And then there's the promoter sequence. And then near the promoter sequence is this operator. So the operator is always between the promoter and these genes. And that's where the repressor protein can bind to. Now, if the repressor protein is inactive, these genes will be on and be expressed, and you'll be building these enzymes that break down lactose. But if the repressor protein is active, these genes are going to be off. So it's the same setup as the TRP operon. But here's the difference. This repressor protein, there's a repressor protein that can bind to this specific operator sequence. And that repressor protein, again, is made by another gene. There's a regulatory gene somewhere else in the DNA that codes for this repressor protein. But here's the big difference. This repressor protein is by default, when it's made, it's by default active, and it's active conformation. So this repressor protein, actually, when it's being expressed, it's active. It's always by default and it's active conformation, which means like once this protein's made, he's ready to go do his job. And his job is to bind to the operator and preventing these genes from being expressed. And so this is the situation we're going to encounter in an inducible operon. An inducible operon is by default off because the repressor protein is by default active, okay, causing these genes to not be transcribed. So this is the opposite of what was happening in the TRP operon. These genes are by default off because the repressor protein is by default active. However, it is possible to turn these genes on. To turn these genes on, though, you're going to have to get the repressor protein to be inactive. And that's going to require a molecule to bind to this protein, this repressor protein, and cause it to change shape into an inactive shape where it can no longer do its job, allowing these genes to now turn on. And so that's exactly what's going to happen in this situation with a lac operon. So that's going to involve a molecule called an inducer. An inducer is a molecule that binds to a repressor protein and causes the repressor protein to become inactive. And so this is basically the opposite of a co-repressor. So a co-repressor, let's compare and contrast here. A co-repressor is a molecule that binds to a repressor protein and activates it, okay, which then causes the genes in the operon to turn off. But an inducer is a molecule that binds to a repressor protein and causes it to become inactive, preventing it from now binding to the operator, now allowing those genes to be turned on. And so that's why we call this an inducible operon. An inducible operon is by default off, but it can be induced. Induced means to make something start happening or to turn on something. Okay, think of like an induced pregnancy. that's the same situation here. This is an operon that's by default off, but we can turn it on when needed. All we need, though, is this inducer molecule to bind to the repressor protein and cause it to become inactive so that he doesn't go and bind to the operator today, so that RNA polymerase, when he binds to the promoter, he can now read the genes and make mRNA, and then the ribosomes can translate that into these proteins. In this case, these proteins are being used to digest lactose. And so in this situation, the inducer is actually a molecule called allolactose. And allolactose is basically lactose. It's just a slightly different version of lactose. And so when there's a bunch of lactose in the environment of the bacteria cell, there's going to be this molecule called allolactose inside the cell. Allolactose is basically it's basically what lactose becomes when it's transported into the bacteria cell. So allolactose, just think of it as lactose. It's this molecule. If there's allolactose, it means that there's a bunch of lactose outside the cell, because when that lactose goes into the cell, it becomes this chemical called allolactose, which is basically still lactose. And so allolactose, which is lactose, is the inducer that can bind to this repressor protein and cause it to become inactive so that this operon can be induced. It can now start turning on. It can start doing what it's supposed to do. These genes can start being transcribed and translated. Now, that makes, if you think about it, that actually is really convenient. So when would we not want these genes on? When would we want these genes off? If you think about it, we would want these genes off if there's no lactose. If there's no lactose, there's no reason for these genes to be on, right? Like why make these enzymes that digest lactose if there is no lactose? So these genes should be by default off. However, when would be a great time for these genes to turn on? A great time for these genes to turn on would be when there's lactose present in the environment, which is a usable source of energy that we can break down into simple sugars and the bacteria cell can use to make energy. And so a great day to turn on these genes would be a day where the bacteria cell wakes up and there's a bunch of lactose, meaning that there's now allolactose inside the cell, which means there's a bunch of lactose today. And that's exactly what happens. When there's a bunch of lactose, it actually acts as the inducer that can bind to this repressor and inactivate it, causing these genes to now turn on. So this is an example, a specific example. The lac operon is a specific example of an inducible operon, a situation where you have a group of genes that are by default off because the repressor protein is by default active, binding to the operator sequence, blocking RNA polymerase. But it can be induced, these genes, this operon can be turned on, these genes can be turned on if there's an inducer molecule that binds to the repressor and inactivates it. And then these genes will now be able to be transcribed and translated, and these genes will turn on, which in this specific situation, it would be great to do that when there's lactose. But when there's no lactose, these genes should be off. So another great example of bacteria cells controlling what genes are on and making sure that genes are on only when they need to be on. And making sure that a group of genes that work together are expressed together. So these genes work together, so it's convenient that they're all sharing a promoter, so that when one of these genes is being expressed, all of these genes are being expressed because they all share the same promoter sequence. So those are two examples of operons, the trip operon and the lac operon. One of them is a repressible operon that's by default on but can be repressed. And one of them's an inducible operon, which is by default off but can be induced, turned on. So we'll compare and contrast those a lot more in class, and because at first it's pretty tricky. They're really easy to mix up, and there's some very specific important differences between them. But it just takes practice thinking about it, and then it's not too bad. So hopefully that kind of makes sense so far. Now the last thing in these notes talks about it talks about positive control of operons. So far we were looking at something called negative control of operons. Negative control is when you have repressor proteins causing genes to be turned off. When those repressor proteins are at least active, right? Like when this guy's active, these genes turn off. When this guy's active, those genes turn off. And so we're looking at negative control of genes, like these TRP genes and the LAC genes being turned off by these repressor proteins when they become active. But there's also situations where genes can be positively controlled in operons. And so I want to show you guys an example of that, because this is also really common in bacteria cells where there's not repressor proteins, but things called activator proteins that can bind to the DNA and increase the transcription and the expression of certain genes. And a lot of times for certain genes, there's repressors and activators involved. And those repressors and activators can be active or inactive. And depending on which repressors are present and active and which activators are present and active, the genes will either be turned on or off, or expressed a lot or expressed a little bit. So it gets really complicated really quickly. But again, we're going to try to simplify this a little bit. So I want to show you guys an example of a positive control, which involves activator proteins. And so here's a picture. Let me just go to this picture here. Of an example in the lac operon, so the operon that I was just talking about, where it is being controlled also by an activator protein. So in the lac operon, the one I was just talking about, there is an operator, right? And that operator can be negatively controlled with a repressor if the repressor is active. But there's also another regulatory gene that can produces an activator protein called CRP. And so CRP, this is a regulatory protein that can bind to a regulatory sequence, so a special sequence of DNA that it can bind to. And when it binds to that sequence of DNA, it actually causes the expression of certain genes to increase, or causes genes to turn on. So it's the opposite of what a repressor is doing. And in the lac operon, there is an example of that, of an activator protein that can help control this set of genes here, the 3 lac genes. And so his name is CRP. And this activator protein is by default inactive. And so when this protein's being made, it folds up into its shape, and it's by default like asleep, doing nothing. It's in its inactive conformation. However, it can change to its active conformation. And when it does change to its active conformation, it will bind to this special sequence, a sequence of DNA, in the kind of near the promoter. Actually, in this case, it shows you as part of the promoter sequence here for the lac gene. So it's binding to a specific sequence of DNA. And this guy actually acts as like a magnet for RNA polymerase. So it helps, it has an affinity for RNA polymerase. It helps attract RNA polymerase to this promoter to start reading this gene. So that's what this guy causes to happen. So when this guy is active and binding to the DNA, and actually is like yelling at RNA polymerase, it's like, here, come bind to this promoter. And it helps RNA polymerase successfully bind to this promoter so that these genes can be expressed. Now, this activator protein is only active when this molecule called CAMP binds to it. CAMP, this is called cyclic AMP, or CAMP for short. We've talked about CAMP before. It's a secondary messenger. We talked about this during cell signaling. It's a common secondary messenger inside of cells. And so what happens is when a bacteria cell does not have a lot of glucose available, so glucose is like the preferred nutrient of bacteria cells and like all cells. All cells like to use glucose to do to make ATP from, because it's a very simple sugar that it can very efficiently use for energy. Now, when glucose is not present, this, like in the environment, there's not glucose present and there's not a lot of glucose inside the cell. That's when the cell, you're going to see a lot of CAMP in the molecule. So think of like the cells like panicking today because there's no glucose. It's like, oh my God, we're going to die. There's no glucose. And there's all these signal transduction pathways taking place. And there's a bunch of secondary messengers like CAMP floating around. And CAMP will be present in a cell when there's not a lot of glucose. When there's not a lot of glucose, there's going to be a lot of CAMP. And when there's a lot of CAMP, it'll bind to this activator protein, which will then bind here and cause the expression of the lac genes to increase. If you think about it, if you don't have a lot of glucose, but let's say there's a lot of lactose, maybe you should be focusing a lot more on lactose. So this is actually kind of beneficial because when you don't have a lot of glucose, you kind of want these lac genes to start being expressed a lot. Like if there's any lactose, let's use the lactose. Instead of dying, let's just use lactose, which is not as preferred, but we can use lactose for energy. It's not as efficient because we have to break it down into simple sugars first and transport it into the cell. It's a little bit more complicated, but we can still survive off of lactose. And so when there's no glucose, there's going to be a lot of CAMP, which activates this activator protein, which binds to these regulatory sequences in the lac operon and causes RNA polymerase to start expressing these genes a lot. Okay. As long as the repressor protein is inactive, which means that there's lactose present. So if there's lactose present, this repressor protein, it'll bind to this repressor protein and make it inactive so that these genes are on. And especially if there's no glucose, this guy will be active as well. So now you have a situation where there's lactose present and there's not a lot of glucose. This would be a perfect time to really kick into high gear the expression of the lac genes. Like there's lactose present, there's no glucose, let's use the lactose as much as possible. That's exactly what's happening in this picture here. Now, if there is a lot of glucose available, so let's say you have a situation where the cell woke up today and there's plenty of glucose, it's a normal day, no need to panic, your preferred energy source is present and readily available, then there's not going to be a lot of CAMP in the cell. So when there's high levels of glucose, CAMP, the secondary messenger, is not going to be present in the cell, and it's not going to bind to this activator protein. And when it doesn't bind to this activator protein is going to be inactive because it's by default inactive, which means he's not going to be doing his job today, which is binding to the promoter sequence and acting as a magnet for RNA polymerase. So he's inactive, and maybe there's lactose present. Okay, so if there's lactose present, the repressor protein would be inactive. And so now you have a situation where the repressor protein is not binding to the operator, but neither is the activator protein. So this would probably lead to a situation where these genes are not really being used a lot, maybe not at all. Some promoters are pretty picky. They have to have these activator proteins for RNA polymerase to come bind to it. That's the situation for sometimes. And for the lac operon, that's kind of the situation here. Like RNA polymerase really doesn't like to bind to this promoter unless the activator is there. So today, he's not really binding to the promoter, and these genes are being expressed very little, or maybe not at all. But if you think about it, that kind of makes sense. Even though there's lactose present today, if there's glucose present, we should still be focusing on glucose. Like even if so it's kind of smart if you think about it. Like glucose is present and lactose is present, which would cause this guy to be inactive and this guy to be inactive. But since there's glucose present, let's focus on getting our energy from glucose, which is the preferred efficient energy source. And we don't really need to focus on lactose right now. Like it's great that it's available today, and we're not going to actively repress these genes, but we're not going to actively activate these genes either because there's glucose present. Now, if there was no glucose present, then we would go back to a situation up here where now the cell is panicking. And there's going to be cyclic AMP, and it's going to activate this activator, and then this activator will cause the expression of these genes to start increasing, right? And now we can start focusing on lactose. But until that happens, like let's maybe just cool it with these genes, even though lactose is present and this repressor is inactive. And so that's what happens here with the lac operon. So this is an example of both negative and positive control kind of determining when these genes are being turned on and how much these genes are being expressed. I hope that this is kind of complicated, especially at first, but I hope that makes a little bit of sense right now. We're going to talk about this a lot in class and do some graphic notes together, and hopefully make this a lot less complicated. So anyway, thank you guys. That's it for this topic. I'll see you guys later.